Hello everyone, welcome to today's experiment. Today we learn gametogenesis. This is a compulsory question which carries 3 marks. 1 mark is for slide identification and 2 marks is for writing comment about the slide. To start with, let us learn what is gametogenesis. The process of formation of haploid gametes in a sexually reproducing organisms from diploid mother cells through meiosis is called as gametogenesis. Gametogenesis are of two types. In males, the formation of male gametes is called as spermatogenesis, where the male gametes are called as sperm. These male gametes are produced in the gonads called as testes. In females, the formation of female gametes is called as oogenesis. The female gamete is called as ova. The formation of ova takes place in ovary. So today, let us study the transverse section of mammalian ovary and mammalian testis. So let us start with the transverse section of mammalian testis. So testis is a part of male reproductive system. A pair of testes is present in male reproductive system. Each testis contains 250 lobules known as testicular lobules. Each lobule contains 2 to 3 coiled tubules called as seminiferous tubules. This seminiferous tubules is lined by germinal epithelium. Germinal epithelium encloses two types of cells. They are spermatogonia and Sertoli cells. Spermatogonial cells undergo spermatogenesis to produce sperms. Spermatogonial cells undergo meiosis and they form primary spermatocyte, secondary spermatocyte, spermatids and spermatozoa. The next type of cell is Sertoli cells. This Sertoli cells is also called as nurse cells, where they provide nutrition to the developing sperm. Between the seminiferous tubules, you observe a connective tissue. This connective tissue contains an interstitial cell called as Leydig cells. The main function of the Leydig cell is to secrete a male sex hormone androgen called as testosterone. So, this is about the transverse section of mammalian testis. While identifying the slide in the microscope, you just have a clue that you observe a compartments same as it is in the diagram. These compartments are called as seminiferous tubules. Keeping this in mind, you can identify this slide as TS of mammalian testis. While identifying, this identification carries 1 mark. Mainly, you have to report it as transverse section of mammalian testis. Don't just write mammalian testis, which don't fetch you 1 marks. You have to mention the section as transverse section of mammalian testis. Now let's see the microscopic view of the transverse section of mammalian testis. Now let us study the transverse section of mammalian ovary. So ovary is a part of female reproductive system where the female gamete ova is produced. This ovary is surrounded by a single layer of germ cell called as germinal epithelium. Ovarian stroma contains two regions. The outer region is called as cortex and the inner region is called as medulla. The outer region cortex contains different ovarian follicles such as primary follicle, secondary follicle, tertiary follicle, Graafian follicle, corpus luteum and corpus albicans. Here, primary follicle and secondary follicle contains primary oocyte. Tertiary follicle contains primary oocyte. Eventually, by meiosis, this primary oocyte is converted into secondary oocyte. Now, the mature follicle is called as Graafian follicle, which contains secondary oocyte and a fluid-filled cavity called as antrum. This Graafian follicle ruptures and releases secondary oocyte, and this process is called as ovulation. The remaining ruptured follicles, they get converted to corpus luteum. The remaining mass of Graafian follicle develops into corpus luteum, which is characterized by a thick yellow mass of cells. This corpus luteum produces a hormone called as progesterone. When the ovum is fertilized, corpus luteum continues to secrete progesterone, which is very important for the implantation. If the ovum is not fertilized, corpus luteum degenerates to form corpus albicans. To identify the microscopic view of transverse section of mammalian ovary, I would suggest you to keep a clue such that you identify a Graafian follicle in your microscopic view. You can see a large circular shape where you can see this fluid-filled cavity, that is antrum, and you could find a dark spot which is identified as secondary oocyte. So if you keep this clue, you can identify the given slide as transverse section of mammalian ovary. And once again, while reporting or while identifying this slide, mention it as transverse section of mammalian ovary.